I'm Nicole.
I'm Jordan.
And I'm Katie.
And this is our ear tour video.
Here we have the auricle, which is also known as the pinna. It is the outermost part of the ear. Down here we have the earlobe, and around the side is the helix, which turns into the crease of the helix. On the inside here we have the scaphoid fossa, and then we have the antehelix, which turns into the crura of the antehelix. In between the crura is the triangular fossa. Down here we have the cymba concha, and down here we have the cavum concha. Right here is the antitragus, and in between is the intertragal notch. Right here on a normal ear would be the tragus.
So Katie just talks about the anatomy of the outer ear, which is also called the auricle or the pinna. So now I'm going to talk about the external auditory meatus, or the external auditory canal, outer ear canal. So the structure of the outer ear helps direct sound towards the concha bowl, which then puts it into the external auditory meatus. And I'm going to turn so you guys can see a better view. So the outer third of the EAM is cartilaginous, and skin in this portion has a lot of glands, and the main ones are sebaceous gland, which secretes sebum, and the main production of these secretions is cerumen, earwax. And so cerumen helps to protect the rest of the ear from bacteria or insects, anything that might get in the ear that we don't want in there. And then the inner 2/3 of the external auditory meatus is the bony portion. And so this enters the tympanic portion of the temporal bone, which is right here. And then these are the mastoid ear cells right in here. The two portions meet at the osteocartilaginous junction. And just to sum up, the main function of the EAM is to protect the tympanic membrane, which is the name for the eardrum, from trauma, and it helps to warm the air before it reaches a tympanic membrane. So it keeps it at a constant temperature and humidity level. And the EAM is really effective at delivering high frequency over low frequency sounds, especially sounds between 2,000 and 7,000 hertz.
So here we have the tympanic membrane. The tympanic membrane, also called the eardrum, is the last stop for the external auditory meatus. The tympanic membrane is comprised of 3 layers: lateral layer, center layer, and medial layer. The lateral layer can be viewed from the EAM and contains epithelial tissues, which is the same tissue of the ear canal. The central layer is fibrous, connective tissue that provides support and strength and also helps the eardrum vibrate for sound. The medial layer is lined with a mucous membrane that leads to the middle ear. The tympanic membrane is extremely thin. This allows the membrane to vibrate according to sound and improves transmission to the inner ear. So now we enter the middle ear. The lateral wall, which is not pictured here, contains the tympanic membrane, which I just talked about, and the squamous portion of the temporal bone. Here we have the superior wall, or the ceiling, and here is the tegmen tympani, and this provides protection for the middle ear and separates it from the meninges. Here we have the inferior wall, or the floor, and this contains the jugular fossa, which houses the jugular vein. And so here we have the anterior wall, and here is the internal carotid artery, and the carotid canal encases it. And then here we have where the cochlear reform process would be, and it contains the tensor tympani muscle. And then also not pictured here is the eustachian tube, which acts to equalize the pressure of the middle ear to that of the external auditory canal. You can also voluntarily open your eustachian tube by swallowing or yawning.
So now I'm going to talk about the medial wall. This is the wall that leads to the inner ear space. So this is the oval window right here, and it's very important for the physiology of hearing because it contains the footplate of the stapes. This is the round window. It has access to the basal turn of the scala tympani of the cochlea. In between those two is the promontory, which is the promontory of the cochlea. So it's shown here on the medial wall, and it's formed by the cochlea's basal turn. This is the prominence of the facial nerve canal. It's the projection formed by the canal that houses the facial nerve. And then this is the prominence of the lateral semicircular canal, which is very important for the vestibular system, which we will be talking about later. And then here is the tympanic nerve that's on the promontory. So now I'll be talking about the posterior wall, which borders the medial wall that I talked about. So the tympanic adductus is the entrance to the tympanic antrum, which is a portion of the epitympanic recess or attic that leads to the mastoid ear cells. So it connects the middle ear space to the mastoid ear cells of the temporal bone. The pyramidal eminence is right here, and it's behind the oval window. It's near the prominence of the facial canal, and inside it is the stapedius muscle. The tendon of the stapes attaches to this. And then the chorda tympani nerve is right here, and it passes through on the border of the medial and the posterior wall, and it pertains to taste and some related activity. And then this is the chorda tympani nerve, which passes through the border of the medial and the posterior wall, and it also joins the lingual branch of the facial nerve, which is right here.
The middle ear is also home to the three smallest bones in the body, the malleus, the incus, and the stapes. The landmarks of the malleus include the head, the neck, the anterior process, the lateral process, and the manubrium of the malleus. On the incus we have the head, the neck, the short process of the incus, the long process of the incus, and the lenticular process of the incus. On the stapes we have the head and the neck, the anterior and posterior crura of the stapes, and the footplate of the stapes. These three bones form a chain which extends from the tympanic membrane to the oval window on the medial wall of the middle ear. The ossicles are held in place by 6 ligaments. The superior malleolar ligament, which attaches the head of the malleus to the tegmen tympani. On the incus here, we have the superior ligament of the incus, which attaches the head of the incus to the tegmen tympani. Right here, we have the anterior malleolar ligament, which extends from the anterior process of the malleus to the anterior wall of the middle ear. Back here, we have the posterior ligament of the incus, which attaches the short process of the incus to the epitympanic recess. Over here would be the lateral malleolar ligament, which attaches the neck of the malleus to the bony wall superior to the tympanic membrane. Down here, we have the annular ligament, which attaches the footplate of the stapes to the oval window. On the stapes, we also have the stapedial tendon, which extends from the pyramidal eminence to the neck of the stapes. Now let's put together all that we have learned up until this point. So far, we have the sound waves traveling through the external auditory meatus, which hits the tympanic membrane, causing it to vibrate. The tympanic membrane articulates with the manubrium of the malleus, which is the beginning of the oscillar chain. This causes the malleus, the incus, and the stapes to be put into motion. The stapes is attached to the oval window via the annular ligament and causes the oval window to move in and out with the movement of the ossicles. Here in the middle ear is where acoustical energy is turned into mechanical energy.
So now we're going to talk about the inner ear, and we're going to start off with the cochlea, which is right here. So vibrations displace the perilymph of the cochlea and enters a bony labyrinth called the scala vestibuli, which is right here. The vibrations continue on in the scala vestibuli up to the heliocatrema, which is up here on the apex. And then it goes back down to the scala tympani, which is right here, and exits the round window. Here we have the scala media, or the cochlear duct, in blue, and that contains endolymph and is the membranous portion of the canals. As the sound vibrations enter the scala vestibuli, the vibrations are also introduced here in the cochlear duct, which causes the Reisner's membrane to vibrate. The endolymph is then displaced. It causes the basilar membrane to move. As the vibrations go down the scala tympani, it causes the basilar membrane to move and stops at the round window.
Within the scala media lies the organ of Corti, which is the end organ of hearing. As the perilymph fluid travels down the scala tympani towards the oval window, it causes the basilar membrane to vibrate, which causes the organ of Corti to be put into action. If a tone is of low frequency, it will cause greatest displacement of the basilar membrane near the apex. If the tone is of high frequency, it will displace the basilar membrane near the base. This is called tonotopic organization. The organ of Corti consists of the tectorial membrane, which moves in the windshield-like fashion. Under the tectorial membrane, we have the inner hair cells and the outer hair cells, the tunnel of Corti. Down here, we have the spiral ganglion and the auditory nerve. The outer hair cells improve and sharpen transmission of sounds to the inner hair cells, which is transferred ultimately to the auditory nerve, which is then transmitted to the brain via the central pathway.
So we talked about the auditory system of the inner ear, and now we're going to be talking about the vestibular system, which pertains to balance. So the vestibule is the entrance to this whole system right here. So it lies medial to the middle ear and it borders with the oval window. So the area is filled with perilymph, and then within this fluid is the membranous labyrinth that houses the utricle and the saccule, which are the end organs. So the saccule is on the medial wall by the cochlea, and the utricle, as you can see, is bigger, and it's continuous with the semicircular canals, which are these three right here, and I'll be talking about them later. So the receptor end organs of the utricle and the saccule are the maculae. So the macula of the utricle responds to horizontal stimulation, but the macula of the saccule responds to vertical stimulation. So when the fluids start moving in the inner ear, it stimulates the vestibular system. This is called the utriculosaccular mechanism, and it processes linear acceleration. So the utricle and saccule are stimulated by the distance rate of change of linear velocity. So how do they do this? The maculae have hair cells that act as biological sensors, and they're innervated by nerve fibers. So the cilia on them are called stereocilia, and they're embedded in an epithelial gelatinous membrane called the otolithic membrane, and on top of this are the otoconia, and they're calcium carbonate crystals. So stimulation happens when linear acceleration shifts the otoconia on the membrane due to inertia. So we talked about how the utricle and the saccule pertain to linear acceleration and their role in the vestibular system. So now let's talk about the three semicircular canals. So we have the anterior, the posterior, and the horizontal. As you can tell, they're all perpendicular to each other, so all spatial dimensions are covered. So the right posterior canal is going to be paired with the left anterior, the right and left lateral canals are paired together, and then the left posterior and the right anterior canals respond together. So if you turn your head, a response will be elicited from the paired structure, and I'll go into that later. So all the canals contain endolymph, and they all return to the utricle, right here, through structures called ampullae. And inside the ampullae are receptor end organs called cristas that help to maintain equilibrium. So the crista contains vestibular hair cells whose stereocilia form the cupula, which is a gelatinous mass. So how does this tie back to the equilibrium in the vestibular system? When the head turns, the endolymph lags behind, which puts pressure on the cupula, and so the stereocilia bend. So when the head is turned, the cupula of the paired canal, remember that was the right posterior with left anterior, right and left lateral, and left posterior and right anterior. So one is going to be excited while the other is inhibited. So semicircular canals, to sum it up, are responsible for angular acceleration, which is measured in degrees. So the outer ear processes acoustic energy, which then travels to the middle ear, which converts this energy into mechanical energy using the impedance matching system. And then the inner ear uses its fluids to help convert these into electrochemical impulses that travel through the auditory nerve and the auditory pathway. And that is how we process the sounds that we hear.